So I've always tried to keep my private and professional life a little bit separated, and I never talked about cleaning before, and now it comes very close. Why? Because I lied for, I think, almost 10 years to my family and friends and my husband, and I told them that I don't know anything about cleaning. And I was so convincing in that lie that actually we outsource these activities at my house. So every Thursday, like today, I leave the door open, somebody comes in, cleans my house, and that's it. Now, if I'm going to talk about cleaning and good cleaning today, that budget for these outsourcing activities might change, and I don't want that. So I really don't want that, and that's why I ask you, so please keep this a secret. I didn't put my name on the slide. That's deliberately done. So if I can trust you, then of course we can continue, because of course the purpose of today is to talk about cleaning validation, and how to design a proper cleaning validation for reusable devices. So remember, it is top secret, yeah? Because not all of you raised your hands, I wanted to give tips and tricks on how to design a good validation. I will give them, but as Ron Brown, he gave you some homework, I'm going to let you work now. So I will not say what the tips and the tricks are. You can find it on the slides. It's very easy. I couldn't practice it with my children because of my lie. But it will be clear. So if you know where is Wally kind of concept, you will be good to go. Now let's start. Cleaning is about a reusable device that is used and you clean it, right? Very simple. You don't need any specific definition. And I'm talking about cleaning. But how do you know when something is clean? I think some of you saw the laboratory yesterday. How do you know when you look at the device that it's clean? You have a lot of different analytical techniques that you can use. We have a whole laboratory, so that's great. If they use it in the hospital and use microscopical techniques, you can really be sure that the device is cleaned. But if you then go to the reality and the clinical side, how do they do it in the hospital? I was a little bit disappointed they used their eyes. It's a very subjective way of demonstrating that something is clean. So you as a manufacturer have to decide that based on this inspection that somebody in the healthcare industry will do, that your device is clean. So you have to assume that what they see as clean is correct. So they cannot make a misunderstanding, and making a wrong assumption can be disappointed and can also be challenging, of course, because cleaning must be thorough. So we have this process, device used, cleaned, reused, and the only control that we have is this, right? So it is crucial that the cleaning procedure has to be validated. I think that is the most important asset. Now, before you start with your validation, one of the things that you have to keep in mind, and the starting point in all of this, is you have to have a good cleaning procedure. You cannot validate anything without a cleaning procedure. Now, this is the cleaning procedure that I used at home. Not to demonstrate that cleaning is very effective, but just to make sure that these activities will be outsourced. So if you have any questions later on how to outsource your personal cleaning activities, this is a good tip and trick. This is not your task to do now, but it works very well until still today. So if you have your procedure, how are you going to validate it? There are 4 main steps. You have your device, you soil it, you clean it, and then you evaluate for your cleaning procedure. Now the task begins. Now the tips are going to show up. So write down. Perhaps I will remove the tips when I distribute the slides, because some of you still not trusting them. It starts with your device, knowing your device. That is crucial. What is the intended use of your device? What are the difficult surface area to clean? That is crucial information that you have to discuss. You as a manufacturer, we as a contract lab need to know that. Can you disassemble your device? Also very important. Do you disassemble it during the cleaning? Yes or no? Important steps. Henry told already this, that a device has to be in simulated use before you start your cleaning process. Why? Because it goes through this process hundreds of times and soil may accumulate. You have to take that into account in a good validation. Is there any guidance on how many cycles you have to use or process it before you start your cleaning validation? No, a rule of thumb, what we use is 6 cycles. This is well accepted by FDA, by authorities. So 6 is the minimum that you have to do before you start your cleaning procedure. And then another thing that Sophie mentioned this morning for the BEP, but in the same context, if you want to do a cleaning validation for your devices, you can also do something as family grouping. If you have a same set series of devices, you want to clean them through the same process, then of course family grouping is an option. So we went from your device and we're going to step 2. Soiling. Soiling is very simple again in my head. You make something dirty. This is something that I can say proudly, I'm very good at it. Soiling is easy, you make it dirty, and that's it. Unfortunately, when I entered into this world of healthcare reprocessing, apparently soiling is not just that simple. First of all, you need to think about your soil components. You cannot just make it dirty with whatever. So you need to know with what kind of soil is your device coming in contact. It can be blood, it can be fat, it can be bone marrow, it can be mucus, saliva, sweat, whatever. Your soil must contain the components that are relevant for its application. If it's irrelevant, it is nonsense. Like Dennis said, garbage in, garbage out. Make your soil relevant. Then how do you soil it? Apparently there are also different types of soiling, and it has to be, again, applicable for clinical use. Take these scissors, for instance. A part of the scissors are in contact with the blood, so you fully immerse them. But the handles of the scissors are not. So therefore you use soiled gloves and contaminate them in such a way. If you make the soil too worst case and overestimate it, you might get into difficulties in your validation process. Soiling can be through spraying, different types of ways, and it is best that it resembles the clinical way, how it is actually used as much as possible, but in a worst case or more extremer conditions. And what's already mentioned, dry time. If you're doing the dishes at home and you are well organized, this is not my situation, but you eat, you do your dishes, it's easy. If you wait until the next day, it's harder to get the spaghetti sauce off it. It's plain simple, but take that into account when you think about soiling. Now we come to the third step, and that's the actual step, and that is cleaning. Cleaning is something very simple, and that is where we are here about, how to validate that cleaning process. So it's only the third step in your validation, and that's the thing you want to validate. Cleaning can be manually, doing the dishes, brushing, flushing. You can use a sonicator, or can be automated in a washer disinfector where you have different programs. You don't need to necessarily have all these steps, but these might be important. So you have an automated and a manual cleaning procedure. What's the difference? I would always choose this one because the machine is doing the work, you don't have to do anything. And if you can possibly validate a manual and one cycle of an automated cleaning, if your device cannot withstand an automated cleaning, then you're good to go with only a manual cleaning procedure. Okay, the cleaning procedure itself, what do you need to do? You have your procedure, and then you have to validate it. So you have your device, it's dirty, and then you follow the procedure. You don't follow the procedure exactly. So for in this procedure, place in hot soapy water, you have to go for a more worst case, a more extremer condition. So take, for instance, cold water in this procedure. To make it more clear, this is an instruction for use, and these are your validation parameters. What you see here is you can use, in the instruction for use, 3 different concentrations of detergent. In your validation, you will use the lowest concentration. Why? Because it's a more extreme version. The lower the detergent, the soap, the less efficient the cleaning. Soaking the device, for instance, 2 to 5 minutes, take only 2 minutes. Just assume that the person that is doing the dishes is in a hurry and needs to speed that process up. And what are the things, how can he search the boundaries of your procedure? Don't go too extreme. It still has to resemble your instruction for use, as a tip that I can give. Detergents, the soap. Can you use any type of soap? What type of soap do I choose? The most relevant thing is choose a detergent that is compatible with your material. If the detergent says that it's not compatible with, for instance, polyethylene and your device is polyethylene, don't use that. The chemical properties of your detergents are also crucial because it will define how well they can remove your soil. Your detergent has to be available in the country where you are going to sell your product on. If they don't have access to the detergent, it's not very useful for you to validate such a procedure. And of course, there are always differences between the expectations between US and the United States. So take that as well into account if you're aiming for that market. So we have our cleaning procedure, our device was soiled, it's cleaned. How to evaluate a good cleaning procedure? What are we going to look for? We're going to look for traces of the soil, but also for traces of the detergent. Now, the evaluation in a validation is done at 3 levels. The first level that we're going to use is, again, the visual inspection. We have to do it because this is how it's done in the healthcare industry. What we have seen, one of the studies that we did was we soiled the device, cleaned the device, and the technician came to us and said, the device looks clean, but it smells bad. It's not in the protocol, it's not in the guidance, but I can tell you, if something has a bad smell, I think you can stop your cleaning. It's not going well. Nobody wants a device that smells bad into the next surgery. So although it's never there, use a little bit of common sense. If it feels strange all of a sudden, if it smells bad, it is not only just a visual inspection. If there is a change, that is a crucial step. The second step that I'm going to discuss is where it comes very close to where I come from originally within Nelson, extractables and leachables. You take your clean device, you're going to extract it under certain extraction conditions and evaluate it. So we're a little bit back. But what are you going to do from an extraction point of view? You're going to use you can use different kind of extraction methods. You can do sonication, manual shaking, orbital shaking. You're going to try to get the potential remaining soil out of your device. Can be many other different things. It's not like in ISO 10993 that it's described what is your extraction method, your extraction temperature. You can choose whatever you want. You have all the flexibility in the world, but the important is that you determine your extraction efficiency for each validation that you do. For each device, for each step that you do, you have to determine the efficiency of the extraction. And that's a big difference, of course, with the extractables and leachables. And then you analyze your extracts. What are you going to look for? You're going to look for different endpoints. Hemoglobin, protein, total organic carbon, carbohydrates. Are you going to select them all? You have to think, what did I put on my device? If you didn't put blood on them, you don't have to evaluate whether hemoglobin is present. That's just irrelevant. That makes sense. What you need to do is to select at least 2 parameters. And for Europe, for some countries, they require that you determine also the bioload reduction. That means that you have added microorganisms to your soil and you want to see how many of them have been removed. What is the acceptance criteria? The TTC concept within cleaning validations, more or less. There is a tier document that says, okay, there are some of them. For proteins, it's lower than 6.4 micrograms per square centimeters, and so on. But I want to point out that these criteria are only there in the guidance for flexible endoscopes, so a very critical device. For all other devices, let's take, for instance, a monitor in the hospital that you have to clean as well. It has to apply to the same criteria than a flexible endoscope. So there is no differentiation at all at the level of cleaning at this moment. I know with discussion of ALPA that there might be a change in that because it's a little bit strange that a simple device has the same acceptance criteria in extractables and leachable. That's not the case. You have different thresholds depending on your device and things like that, and contact time. Here, this is unfortunately not yet the case. Every good validation, every good procedure also needs its control. When you validate, you must be sure that what you're validating and the result that you come out, and if you want to claim that your cleaning is successful, you have to have a positive device control taken into account and a negative device control. The positive device control is a device that is soiled, not cleaned, and is also used to determine the extraction efficiency. The negative device control is unsoiled, clean, and is used as a test control. You cannot see anything on that because there was no soil on it, to make sure that you take the right conclusions. It's in every experiment that you have to take controls into account. So you have 2 levels already. The third level is about detergents residues. So I've looked for traces of the soil, but traces of the detergent can be toxic to the patient, potentially. So you want to remove that. How do we determine whether there are still residues of the detergents on your device? We do a cytotox test. That is simply the requirement at this moment, and this is how we're doing it. Now, yesterday, Henry told it, I think a lot of other people mentioned it as well, but there are things we have to consider. When you think about devices in general, we know that there is the aging of the device over the life cycle of the device. It's in ISO 10993-1. It's even mentioned in Annex 1 of the MDR. The life cycle of a device changes over time, just by standing on the shelf life and also by being reprocessed. So you can see that perhaps on the surface it's not visually there. We had the example of the tubing that felt harder. There is something that is different. Now, what you can wonder is, can the biocompatibility of a device be changed through cleaning by contact with a detergent? Can the material properties and the material function change, and as a consequence, can the biocompatibility change? When I first started here at Nelson, they always explained me, Pete, Anja, if you think about extractables and leachables, there's always a gap. I'm not sure whether we mentioned it enough. Extractables are the compounds that can potentially come out under worst case conditions. The leachables are the compounds that leach into your body, right? There is a gap. The gap in the pharma industry comes because the drug changes the profile. But can the profile be altered here by aging of the material, by the contact of a detergent? The detergent is a chemical compound that comes in contact with the material. It alternates the material. The processing steps, like cleaning, it's at high temperature. There might be a change. It is not something that, yes, it changes over time. You're really subjecting these devices over and over again to aggressive conditions. And there are people that say, okay, you can use the cytotox test to evaluate whether the device is still biocompatible after the cleaning. But then I have to ask to Tor and other biocomp experts, can you put so much weight on just one simple cytotox test to claim that the overall biocompatibility of that device is okay? I'm not sure. The only thing that I would like to suggest is for people that are in that field of biocompatibility, when you think about your BEP, think about the life cycle. People change over time. Relationships change over time. They don't change for the good or the bad. You can be still happily married after your first nice date, sitting in the sofa like this. You still can be compatible, but can you really prove it? Is it something to take into account? Some thought, perhaps for the Q&A later. That brings me also to the summary of the presentation. We went through the cleaning process. Devices used, clean. The importance of the visual inspection was pointed out. I discussed the 4 steps, the 4 important steps of the cleaning procedure. If you have paid attention, did your work that I asked you for, you have gathered 2 tips for device, 1 for the soiling, 3 for cleaning, 5 for evaluation, and I gave you also some things to consider about, and also to our biocomp experts. And with that, the only thing that I ask you, I didn't tell this. Thank you.